Hi everyone, and welcome to lab number 12, which today focuses on GMO PCR experiment that you would have done in lab. Now you're all already experts at PCR thanks to the previous lesson, but now we're going to use it to kind of see another application of this technology. Keep in mind, you are getting close. You're almost done with this semester. So now the first thing I have to, kind of ask you is, well, what does GMO mean? And that's genetically modified organisms. And today we're going to focus on how that applies to genetically modified food specifically. Now, when we say genetically modified foods, okay, that basically means that these foods are made from at least one plant product that has been modified with foreign genes within it, okay? And a lot of times what we mean by foreign genetic material is that these genes or that piece of DNA in that plant came from either another plant, or it could be genes from an animal or a fungi or even bacteria. Okay, so it just means foreign genetic material, meaning, not originally from that exact plant. Okay, and ultimately, as you already know from our transformation experiments, when you are genetically modifying things, meaning adding extra foreign genetic material, the reason you're doing that usually is to code for a particularly advantageous protein. Okay, now we focused on advantageous proteins for bacteria, which are things like antibiotic resistance. But with modifying plants and crops, there are a lot of different examples of why you would want to make these modifications or what types of proteins you would want to choose. Now, ultimately, when we genetically modify foods, a lot of times you just have to think about what would you want for crops to kind of help society with that particular food. So some of the things that we do is we add pest resistance to give it its own ability to have, pesticide capabilities to kill any potential pests, like things like worms that would try and eat the crop. They add genes for improved tolerance to herbicides, okay, to help better strengthen that plant. They do things to help with the fruit or the vegetable that's coming from that plant. So things like delayed ripening or being able to make more fruits. So for instance, if you're picturing a tomato plant, you might modify it with genes that help it produce numerically more tomatoes. So instead of usually having 10 tomatoes for that plant, it'll be able to yield 100 for that crop. Or to yield bigger, more robust fruits and vegetables, okay? And this is especially important in third world countries where we can kind of help have the crops not only produce bigger, more robust fruits and vegetables, so you are getting more fruits and more food products from that small crop, but also to increase nutrient content, so adding vitamins and minerals that are highly deficient in some of these places. Now, when we think about genetic modifications, there are 5 main steps that I want you to know for how we would genetically modify crops. Okay, the first step that you would want to do if you're genetically modifying a crop is find a beneficial protein. Okay, once you have determined, what protein you want to use, then you can move forward. Now, when I say find a beneficial protein, a big example of this, for instance, is when they found the delta endotoxin, a protein produced from a gene of bacteria known as Bacillus thuringiensis. And with this bacillus protein, this particular toxin can kill specifically corn borer and silkworms without harming other organisms. Now, you may say, well, why is that significant? Well, those are big common pests for crops. So if a plant is able to produce its own natural endotoxin, and I know it sounds funny me saying natural when this is genetically modified, but this is a naturally occurring protein of bacteria, so that's why I use that term. So if you could get the plant to produce this bacterial protein that kills silkworms and corn borers, well, now you no longer need to apply harmful chemical pesticides. You don't have to constantly spray and expose the rest of the environment to the various chemicals that they formerly used on those crops. Okay, so it allows the plant to reduce the amount of crop damage or crop loss from pests by producing its own toxins that are specific to those particular worms or organisms that are known not to harm humans, for instance, or other organisms. Now, once you have identified that particular protein that you want, well, think about it. How do we get proteins? They're initially from genes that then code for RNA, that then code for that protein. So you can't just shove, the protein into that plant cell. You need to be able to go at the base floor of the operation, you can call it. You need to get a gene that then codes for that protein within that plant. So what they end up doing is they isolate or clone that gene from its original organism, and they localize it within the genome and copy it into plasmid, right? We've seen this idea before. This slide is basically showing that step 2 of genetically modifying crops is the molecular cloning that we've talked about previously, where you take a gene of interest from whichever organism you know that it's found in their genome. You cut that particular gene out, put it in a plasmid, and get bacteria to make a ton of it for you. And then you can use those copies of that gene toward the next steps of your genetic modification process. Now, the next step in terms of once you know exactly which gene you're going to use is you have to engineer that gene a little bit. You got to play around with it a little bit, because we just said that a lot of times this gene is coming from another organism. And each organism, especially if it's, for instance, a eukaryote versus a prokaryote, each organism has slightly different gene signals. Okay, so that means that you will have to modify that gene so that it works in plant cells specifically. Okay, otherwise it's not going to end up producing your protein properly once you put it into your plant that you're trying to modify. So what they end up doing are things like removing any unnecessary introns, and what they also do is make sure that it has proper plant gene signals. Specifically, they put a promoter and a terminator sequence around that gene, sequences that are known to be able to be read in a plant cell, okay, that all plant cells will be able to detect and properly, work with. What they commonly use are the CAM-V35S promoter and the NOS terminator. Okay, now you have to remind yourself what these kind of words mean. So first off, promoter, that's the sequence that you want any gene that's going to be expressed to have in order for RNA polymerase to basically properly identify that gene, dock there, and start transcribing a gene. Whereas the terminator is then how we stop the expression process. Now, the ones that they use here, CAM-V, that is from cauliflower mosaic virus, which is known to infect plants, which means that the sequences here are known to naturally start transcription in any kind of plant cell. Then the NOS terminator, that stands for nopalene synthase terminator, and that's from Agrobacterium tumefaciens, which you already know is an organism that can infect plant cells. And if it's able to infect plant cells, that means that this terminator is also a sequence that is properly able to be read in any kind of plant cell. So this means that one or both of these sequences, the CAM-V35S promoter and the NOS terminator, okay, are in the majority of genetically modified crops. That's significant. I want you to circle star highlight the fact that these two sequences are in the majority of GMO crops that we make. That's significant for our experiment today because that means that we can specifically target and identify whether a particular plant contains these two sequences. And if a plant or a food product that comes from a plant has either of these sequences, which are not naturally found in plants. Notice both of them are from organisms that infect plants, but are not plants themselves. One is from a virus and one's from a bacteria. So these sequences are not naturally found in plants. If you can detect them with your kit and your PCR, that means that that food that you're testing or that plant that you're testing has been genetically modified. It has GMO parts. Okay, so really make sure you're very comfortable with that concept of the CAM-V promoter and the NOS terminator being from organisms that infect plants but are not plants, and that these sequences are found in most GM crops, genetically modified crops, because this is the basis of our experiment today. Okay, so now at this point, you have identified what beneficial protein you want, and you have located and isolated that gene, and you have properly modified that gene so that it can work in a plant. So now that leads us to step 4, which is to actually get that gene into a plant. So what they do is they remove cells from a parent plant and they grow it on a special media. And when grown on that special media, it creates what's called a callus. And I want you to circle star highlight that word callus and note that that's basically a clump of undifferentiated cells. Okay, so that's going to now serve as the foundation of getting the new plant. And now what you can do with those undifferentiated cells is transfer your modified gene into that clump of undifferentiated cells. Now that, poses a challenge, because if you think about plant cells, what do you know about them? Well, they have a really tough cell wall, right? So it's going to be tough to just get, whatever you want into those cells. So you need special ways to get your engineered gene into those undifferentiated plant cells, into that callus. And so now we're going to look at 3 ways that they do this. So now the 3 ways that we can get these genetic modifications into the plant cell. The first one you've probably heard before, okay, you usually cover that in lecture, and we're going to talk more about it in the next lab, is the Agrobacterium tumefaciens. And this is bacteria that infects plant cells, okay? So you can use this organism that is known to infect and be able to replicate in plant cells, replicate its DNA in plant cells. You can use this in your experiments, because in nature, this organism has the ability to insert some of its DNA into a host plant's genome. And so you're able to put the genes you want into that piece of DNA, and this organism will insert it into the host plant's genome, okay? And when we look at this in the next lab, you're going to see that whenever you see those giant growths on tree trunks, the big clumps that are basically tree cancer, that's an infection with Agrobacterium tumefaciens. The next way that you could get the modified genes into the plant cell is to use electroporation. And electroporation basically uses an electric current to create pores in the outer membrane of the cell so that the engineered DNA can then enter. So it uses an electric current to force DNA into the plant cells. The third one that I want you to know is the gene gun, and this is my personal favorite because basically what this does is it shoots gold particles coated with the engineered DNA into plant cells. And it's my favorite one because I picture the original scientist who had to think of, hey, you know, I need DNA into a plant cell. What can I do? And his solution was shoot it with a gun. Okay, so I always found that option as quite amazing that someone actually came up with that in the lab, but hey, it works. So I basically want you to remember these three examples and make sure that you know what each one actually is doing. Okay, so make sure you rewind and jot down those notes of what each one actually is. Now please keep in mind that with any of these, none of them is going to be perfect. You actually won't get a lot of DNA that successfully goes into plant cells. So you basically, just like when we talked about in transformation, need to be able to quickly identify and detect which of those few cells actually got the target DNA into it, you know, your plasmid and your modified genes. So what can we use to detect whether or not you got that plasmid in or that the modified genes? Well, you already are experts at that answer, which is use selectable markers. So a lot of times they'll use selectable markers just like we did in transformation. Things like antibiotic resistance or visual color changes like we used. Sometimes they use also fluorescence using GFP. Now, once you isolate exactly which cells were successfully modified, then what they end up doing is they induce those cells with plant hormones, okay? Because remember, those were undifferentiated cells. So now with plant hormones, those cells will be able to differentiate and grow into a full plant. So now that brings us to the last of our 5 steps, which is to backcross our newly grown engineered plant into whatever is the current high yield crop. Keep in mind this can take years because, you know, when you are crossing two organisms, you know, a mother and a father organism, you're only getting 50% of that high yield parent. Okay, the other 50% is the genetically modified one. So basically you want to breed your new genetically modified crop into the currently existing crop so that you get the maximum, you know, best outcome of the crop. And keep in mind on this slide, I like this picture because it shows our previous steps as well. And so you can kind of visualize and really remember the idea of, you know, the plant cells becoming the callus and then selecting for and growing your new plant and then cross-pollinating or back-crossing that genetically modified organism with the previous crop to kind of really maximize your farmland. Now it's time for our Remind app question. What I'd like you to do, since, you know, GMO is such a hot topic still in terms of, you know, controversy, those who support it, those who are against it, those who can see both sides of that argument, which is always very important that you educate yourself on both sides rather than just, you know, looking at a concept and only sticking to one side of it and not hearing or listening to the other side's points. So what I want you to do in the Remind app is for genetically modifying food, I want you to send me one pro. So what's one good thing about genetically modifying food and what's one potential con? What's one potential bad thing about genetically modifying food? Okay, so think about what's good, what can help us and benefit us by using GMOs, and what might be a problem with these modifications. Okay, make sure to send me that in the reminder. Now we get to our particular experiment now that you understand the background information. And so with our experiment, we would be doing GMO PCR that would be testing for a food sample. So you would pick a food sample such as Doritos or Lay's potato chips are usually popular ones that students want to test. And sometimes we do things like oatmeals and whatnot. Whatever food sample you would want to test, we would then test to see whether or not we detect any genetic modification. So you wouldn't know the exact genetic modification. You would just be able to identify, whether or not this food product contains some genetically modified plant products. Okay, and in order to do this, you would use PCR primers for on your test food. So I'm going to keep using the example of Doritos because the chips they use in the pictures usually look like, tortillas or Doritos, and students always love testing Doritos. So you would take your test food and you would use PCR primers that target the promoter and terminator that we mentioned earlier that are added to genetically modified foods. Okay, so these PCR primers would only bind to and amplify the 35S promoter we talked about and the NOS terminator. So you will only see a DNA band for those genetically modified sequences if that food product contains them. If they have not been genetically modified, then the primers have no DNA to bind to. They won't recognize or match any sequence, and you won't see any amplification on your final gel that you do gel electrophoresis with. Okay, now these primers end up giving a product that is around 200 base pairs. So if your test food has genetically modified food within it, then your PCR primers for this set of primers, the genetically modified sequences, will result in a band around 200 base pairs. If there is no GMO in that food, you will see no band. So it's either no band or a 200 base pair sized band, okay, with those primers. Now you will also use on that same test food control primers to make sure that your PCR is actually working. And so the control primers are plant primers that target a chloroplast gene. Usually it's for photosystem number 2, but you don't have to worry about the details of that. Just know that the control primers that you would use target a chloroplast gene that all plants have. So all food that contains plant product, any plant product, would have these sequences, and they would yield a product that's around 400 base pairs. Okay, so every food sample that you test, because you would always be testing, just about everything we test has corn or wheat or some sort of plant product in it. So any food sample that you would have tested in this experiment would have plant product in it. The control primers would recognize that and would give a 400 base pair product. Okay, it's actually 455, but you don't have to worry about that. Just figure around 400 base pairs. Okay, and that would show you that your PCR actually worked. So if it's a non-GM food, okay, it's not GMO, then you would have a 400 base pair product for the plant primers, and you would have no band for the GMO primers. Whereas if it is GMO containing, that food product, such as Doritos would end up with a 400 base pair band of DNA for the plant primers, because it contains corn and wheat and other plant products, and it would have a 200 base pair band for the GM primers, because the genetically modified food has those two sequences. Okay? At the same time, you would also have control foods. So not just control primers, but you would have control foods you would have a known non-GMO sample that you would prepare, and you would also have a GMO positive sample that is known to contain that primer and terminator, so that you can then compare your test food results to those two samples. Okay, if your test food bands match the non-GMO, then you know you have no GMO. And if your test food bands match the GMO positive, then you know that you have GMO in that test food. Okay? And like I said, the key point is that you would then analyze your PCR products with gel electrophoresis and look for those bands. Does it have the 400 base pair band? And more importantly, does it have the 200 base pair band? This is just so that you can visualize what you would have done. There would have been 3 parts to this experiment. The first part is to actually extract and prepare your food sample. So you would prepare your non-GMO and your test samples, such as the Doritos, the oatmeal, the Lay's, whichever one you were using. You would grind it up and isolate and extract the DNA from that. Then the second part would have been the PCR preparation. So first part is preparing the food samples to isolate the DNA, and then the second part would be to set up the PCR. Now I'm just going to switch over to the pen just so that you can remind yourself that when setting up this PCR, you would have had 3 sets of DNA you'd be working with. You would have your test food, okay, that is your experimental group. So your experimental group. You would then also have the non-GMO DNA, which would be your negative control. And then you would have your GMO positive, which, as the name suggests, would be your positive control DNA. And for each one of those 3 samples, your negative control, your experimental sample, and your positive control, you would have 2 different primer sets that you would use. So you would be setting up 2 tubes per sample, one with the plant primers, which are your control primers, and a second tube with your GMO master mix. That's what MM stands for, is master mix, which we covered in the previous lesson, what that would include. This would be your test primers. Okay, so everyone would end up with 6 different PCR tubes to make sure that you have thoroughly and properly set up all necessary controls. And then you notice at the end it would all go into the thermal cycler, because remember that's an important piece of PCR that a lot of students forget. They remember all the master mix ingredients and the DNA template, and they forget that without a thermal cycler, none of that would work. Okay? And that would bring us to the last part of our experiment, which would have been gel electrophoresis. So you would have to do 3 parts to this experiment. The first part was isolating DNA from test samples from food. The second part would be PCR, setting up a PCR. And the third part would be running the PCR samples, the final products, on a DNA gel, an agarose gel, and looking to analyze the bands that we just discussed in the previous slides. Now we get to our results, and just like we did with the crime scene, I have the actual gel with someone wearing gloves and holding it, and then I have the printed pictures for you to kind of help visualize the results. Okay, notice that instead of doing 2 separate gels, they ran them both on the same gel, which you can do if you set up wells at the top and the middle and you stop the gel nice and early, okay, making sure they don't cross into each other. This top part is test food A, This bottom part is test food B. So with these results, basically, like I said before, each student would have had a different test food. So for instance, some students would test chips, some might test oatmeal or food products like that. Now that means that each student gets their own gel here. So when I ask you to analyze these results, keep in mind test food A gel versus test food B gel. Those are two separate students, two separate test foods. So for instance, if you want, you could write chips for test food A and then oatmeal for test food B. And you could even use real names like I did before, Doritos chips versus Quaker oatmeal, whatever you want. And then when you analyze, keep in mind, each gel is loaded the same way. And you have the number for the lane. So this is lane 1, lane 2, 3, 4. This is then again lane 1, 2, 3, 4, etc. And down here tells you what's loaded in each one. So for both of these gels, test food A and for test food B, the non-GMO food with the plant primers, remember plant primers expecting around 400, 455 band, that would be in lane 1. Then non-GMO with GMO primers would be in lane 2 for both of these. Then when you get to lanes 3 and 4, that's where the test food is. So that's what would be different on each of these gels, okay, lane 3 and 4. This one, for instance, like I said, might be chips, and this one might be your oatmeal sample. Okay, and some students would pick oatmeal or chips from Trader Joe's or health food stores like Whole Foods to kind of test the so-called organic non-GMO labels. Okay, so keep in mind, again, lanes 3 and 4 is what's differing on both of these gels where students would have tested two different food products. Okay, if you have any trouble analyzing these, again, relying on lane 7, which you already know every gel must have a ladder, standard, sometimes they call it a ruler. That is your DNA ladder, lane 7 on both of these. If you have any trouble when you're trying to analyze these, just send me a message in the Remind app, and I'm always happy to help. That is it for this lesson, okay? Again, it's kind of calling upon some of the things we worked with before, so the concept shouldn't be as difficult now that you are getting to be experts at this point in a lot of the genetic and molecular concepts. If you have any questions, as always, please send me a message in the Remind app. Make sure you send me the answer to today's Remind app question as well, and please make sure to do the post lab. Thank you and have a wonderful day.